All right, we are going to get going here. I just have to say that it's an amazing treat to have Julia here, who is amazing in really doing research that advances our basic understanding of ecology and evolution, and in the meantime impacts the way we look at weeds from a genetic and evolutionary perspective. So she really does bridge this thing from the very basic theoretical understanding of what's going on in evolution at that level, and also understanding in our cropping systems and what's been going on for the last 100 or so years in terms of how plants are adapting to us as we adapt to them and so on. And she's really interesting in that you could probably find her at an evolution meeting, meeting of the American naturalists and molecular biology meetings, but you could also find her at a weed science meeting. And if you know the history of NC State and that interfaces between genetics and application, she fits in just perfectly. So she's going to come here next year. No, no, but she is going to tell us about what she does and how that relates. So I'm not going to go through all of your awards and everything like that. I just want you to get up there.
Thanks, Fred. Thanks so much for joining today. It's amazing to see so many people here, and I've had such a nice time chatting with people this morning, going for a hike with Fred yesterday. It's been a nice visit. So yeah, I'm excited to tell you about some of the work I've been doing throughout my career, and now what my lab is doing. Just started this summer at the University of Chicago. I'm kind of pertaining to this classic question in evolution, the tempo and mode of adaptation, but thinking about it on microevolutionary timescales in response to pretty rapid change that's occurred across the landscape. And that's land use change and management change as it pertains to agriculture. So, I think a lot of us in this room can recognize the impacts that humans have had on reshaping our environments, but not only our environments, but the organisms that live in them, right? And so in ecology and evolution, some of our strongest examples in evolutionary biology come from these human-mediated selective pressures. That might have been industrializations in the 1900s, driving selection for these better camouflaged peppered moss on these forest trees. It might have been overfishing on Atlantic cod off the coast of Canada, drive rapid life history evolution, or even more recently, the evolution of tusklessness in African elephants as a result of the ivory trade. So these are all, I think, really striking examples, populations and species that have adapted in the face of environmental change. But I think we're increasingly aware that this is not going to be the case for all such populations and species. So I think an important question is really understanding what determines whether and how quickly populations can adapt to environmental change. So an important way we can get at this is turning to a framework at the intersection of ecology and evolution, and that's evolutionary rescue theory, right? And that posits that's not just the genetics of populations that determine the likelihood of avoiding extinction or undergoing evolutionary rescue, but it also relates to the demography of populations. So we can imagine some environmental change occurs here when the x-axis is at 0, and that population was not suited to that environment. So you're gonna, a lot of individuals will die. You'll undergo kind of an increasing risk of extinction as populations die off, or individuals die off in that population. But a rescuing mutation might arise, in this case, a new mutation that allows this population to go demographic, recovery, and local adaptation. But this is actually a pretty slow way in which populations can evolve because of all of this waiting time for these new mutations to arise. On the other hand, in larger populations where there might be more individuals for which new mutations can arise on and persist in these populations even before the onset of environmental change, we might have adaptation occur much faster and even avoid any of that kind of extinction risk through drawing on these standing genetic variants. And increasingly, since some seminal review, we have seen standing variation play a really important role in rapid adaptation. So when we're thinking about whether populations will adapt to environmental change, I think a really striking framework for doing so is in response to the agriculturalization of our landscape, which has occurred extremely rapidly over the last 2 centuries in particular. Right, so in the 1600s across the United States and Canada, we were only actually using 1 million hectares of land towards crop yield, crop production. But this is, of course, rapidly expanded and increased, especially since the 1800s. And this really occurred along with increasing population size and crops growth, right? So this really started with the Industrial Revolution, which took more than 50% of North America's workforce out of agriculture and into other industry, right? And such that monoculture cropping and industrialization of agriculture was really growing along this time period. But at some point, we realized we couldn't just increase the amount of land we were using to grow more crops, but we had to figure out other ways to increase the efficacy of these practices. And so in the 1950s, you see the slope start to decrease, but crop yield was still increasing at this time, right? So a lot of people have deemed this the Green Revolution. This really started with an evolutionary innovation, right? And that was the breeding of high yield crop varieties. But along with that came highly controlled irrigation, widespread mechanization, and increased, and some would say, over-reliance on chemical inputs like fertilizers, pesticides, and herbicides. So while this really increased the efficacy of agricultural practices, it also really intensified the selective pressures occurring across these landscapes. And I think the strongest kind of illustration of that is the rapid evolution of resistance in response to herbicides. So since herbicides were first introduced across the landscape in the 1950s, we've seen evolution evolve in a number of species. So this figure probably outdated at this point, but not just across hundreds of species, but in response to every chemical compound of herbicide we've been able to innovate. So obviously this is a huge economic importance. These herbicide-resistant weeds are competing with crops for resources, and so they're drawing down crop yield, posing huge economic costs. Right, and so because of that, the field of weed science has done a lot of work to figure out what are the genes targeted by these herbicides, but not only that, what are the particular amino acids and non-synonymous mutations within them that actually confer resistance. So a lot of these herbicides we actually know causal genes and causal SNPs, but we didn't really understand kind of the evolutionary dynamics and history of these resistance mutations across the landscape and how they actually have been responding since the introduction of herbicides. And actually, there's more complexity than just SNPs, and I'm not going to focus too much on this today, but we also know that there's gene amplifications and even polygenic architectures, so SNPs all across the genome that can confer resistance in species. And so some of my work has been to kind of uncover those other architectures of resistance. So today I want to focus on two kind of related works, but to understand kind of the tempo at which the genetics of these populations have actually responded to this environmental change. And then also thinking about the genetic modes of the standing variation or new mutation and how that might have played a role in the spread of resistance across the landscape. So to do so, we've been really focusing on this problematic agricultural weed, Amaranthus tuberculatus. And maybe some of you have seen this around. It's actually here in North Carolina, but it's especially a huge issue all across the Midwestern United States. I call it problematic, but really this is a native species, right? And a lot of people wouldn't necessarily think of a native species kind of being invasive, but that's definitely the case. This species predated the agriculturalization of its landscape. So there's actually two subspecies that were at one point historically reproductively isolated on either side of the Mississippi River. So there's lots of structure and where diversity is found. It's also dioecious and wind pollinated and diploid. So this is actually a pretty rare combination in plants in that most annual plants are selfing. So here's one where male and female flowers are actually found on separate individuals. Before I get into and so, we've been working on developing the system to really understand rapid adaptation, agriculture, and herbicide resistance evolution. And it's really been a collaborative effort. So throughout my PhD with Stephen Wright and Jonathan Strom at University of Toronto, we were working to collaborate with weed scientists, Patrick Trinell in particular, who've been working on water hemp in these agricultural systems for 30 years before we started, thinking about the genomics of it. Collaborating with Detlef Weigel to develop some of these resources, and in my postdoc, Ali Otto, to think about modeling kind of the mechanisms and processes that were driving some of these patterns we were seeing across the genome. Before I get into too much agricultural adaptation, one of the things I just want to emphasize that all of the variation we see at the phenotypic level is really structured by this history strategy, separate sexes. And so we've done some recent work to understand the architecture of separate sexes. But even there, this species is extremely diverse in that we actually see some gene exchange between males and females in the sex chromosome that also is reflected by leakiness in the expression of sex. So while it's been called a dioecious species, we still see male individuals that have female flowers and vice versa. So just one more illustration that you're going to see today of how diverse this species actually is. But let's get back to this main question. How and how quickly has water hemp actually adapted to agricultural change? So to look at this, we really leverage the fact that this is, like I said, and is a native species that predated a lot of the agricultural change across the landscape. And that ancestral habitat is still there, right? We have these non-agricultural populations, these natural populations in riparian settings where we can still find populations of water hemp. So I went out and did a nice road trip where I did these pairwise collections of natural and agricultural populations across the range so that we can kind of disentangle what are the broad scale drivers of diversity from what are the consistent shifts that have had to occur in this plant's invasion into agriculture. So we've done some phenotyping to look at in a common garden what traits are differentiated. We've published that work. I'm not going to talk too much about it today. Since I'm at the Genetics and Genomics Institute, I'm really going to be focusing on some of the genomic results. But before I get there, I guess, to talk about the ecology and environmental settings in which these populations are found, here's just an illustration. So a natural population in southern Illinois here, pretty sparse populations, individuals like 30 centimeters tall, occurring right alongside this lake along the beach, but less than 3 kilometers away, a soy field just a couple weeks out from harvest, completely overran by many meter tall water hemp. So very, very different types of selective environmental pressures occurring here. So after this phenotyping experiment, which I didn't tell you about, one of the first things we did was to look at the genomic signatures of adaptation to agriculture by doing whole genome sequencing about 200 individuals from across these collections, right? So one thing we might be interested in, well, just asking how different are the individuals that occur in these different habitats across the genome. So one way of doing that is calculating FST, which is a statistic that reflects genetic differentiation. So a value of 1 means that variants are almost completely unique across those two habitats. But we're seeing a value approaching 0.00075, which tells us that most of the variants are found at almost equal frequencies across both habitats. So what that tells us is that there's a really important role of gene exchange in shaping variation across these populations. And I think, while simple, this is really important in and of itself, because it tells us that these selective pressures and the management regimes that we're implying in agriculture actually have a really important effect on shaping genetic and phenotypic diversity in nearby natural populations. So while there's lots of evidence for gene flow, we still know that selective pressures are differing between these habitats, like herbicides, right, is one of the first things we might think of. And that was one of the first things I thought of too. And so we know these causal resistance alleles, so we could just say, well, how differentiated are those particular alleles, right? Not just these genome-wide average values. So one of these alleles is a deletion of an amino acid in this gene PPO. It's a target of PPO inhibiting herbicide. And remember, we have this paired design. So what we might expect is that there might be differences in the frequency of resistance, where if they're costly to be at lower frequencies in natural populations, especially in the face of gene flow, that might be continually introducing them outside of agriculture into natural populations. And indeed, that's what we find. So it seems like resistance is a source or something in which antagonistic selection is acting on in these populations. But that's just for one resistance allele. What was pretty surprising to see is not just the high frequency of resistance in some of these natural populations, but the extent to which they're, sometimes even at higher frequency in their nearest agricultural population. So this was one way in which we can think about how consistent the costs of resistance alleles actually are. And I think it tells us that they're not actually very consistent at all. But how to contextualize this in terms of how important or how strong is antagonistic selection on resistance across the environments compared to other sites or other sources of selection across the genome? So we can perform a test that does exactly that, and leveraging our paired design, we can ask if there are particular SNPs that are more frequently found in one habitat, right? Alleles that are only ever found in agricultural habitats. So it's similar to an FST type test, but we're leveraging this structured paired design. And so we do this test at every site across the genome. And on the y-axis here is just the Cochrane-Mantel-Hansel test is just the generalized version of this. And the larger value means that there's a SNP that has a stronger evidence of playing a role in agricultural adaptation. Again, just to orient you, we do this test across 16 chromosomes. And what we find actually highlighted in red is a set of alleles that even after multiple test correction play a really strong role in adaptation to agriculture. Collectively, these alleles in red are enriched for functions in response to growth and reproduction, but also tolerance to drought, stress, and herbicides. And actually, the gene with the highest, strongest evidence of playing a role in adaptation to agriculture is actually PPO, right? The target of PPO inhibiting herbicides. So, it's kind of nice because it tells us genomics actually works. We can do these designs and recover signatures of selection, which we expect to be quite important. But I think, again, it also just emphasizes how important herbicides actually are in shaping fitness in these agricultural and natural habitats. So, population genomics, I think, is really exciting in a lot of ways. We're generating a lot of hypotheses about what genes might be important here for fitness and for adaptive traits. But I think it's also nice because it provides us a set of alleles that we might further investigate to understand their evolutionary history. In particular, address this question: what are the timescales and processes that have actually shaped these patterns that we're seeing across contemporary genomes? And so it's really exciting that we could turn to time series data that's actually contained within herbarium to test this question. So the contemporary samples I'm highlighting here in light gray and dark gray, but I was actually able to curate about 108 herbarium samples by working with 8 different herbarium curators, both university and independent herbarium, to sample and sequence those that span not just this geographic range, but data all the way back to 1828, right? So some of this during this expansion period, but definitely transitioning through kind of the intensification of agriculture as well. And because of the care, really remarkable care that both community scientists and botanists put into annotating these sheets, we could also actually say something about the environments in which those samples were collected on. So it's not just how have these, agriculturally adaptive alleles change through time, but how might that be mediated by differences in these selective pressures across habitats? So I'm not the first person to think about, what processes might contribute to agricultural adaptation and over what timescales, even in Amaranthus. Actually, there was a really nice botanist, Jonathan Sawyer, in the 1950s, who used some of these very same herbarium samples to delimit these 2 subspecies. So Amaranthus tuberculatus var. Rudis, referring to its ruderal weedy nature in the southwest, and var. Tuberculatus to the northeast. And you'll notice this northeastern subspecies range actually very closely mimics the major Mississippi and Missouri water basins. So he hypothesized this was a much more riparian adapted subspecies, and actually that it was the expansion of this southwestern type northeastwards that led to a lot of the agricultural invasion we're experiencing today. Right, so Sawyer was effectively making a hypothesis about the role of standing variation in var. Rudis in rapid adaptation to agriculture. So we can test this Sawyer's hypothesis with our herbarium data, right? We can actually ask how the distribution of ancestry not only has changed through space, right, from on an axis of west to east in our sampling, but also through time in our herbarium samples. So what I'm going to show you is each dot is an individual or a sample in which we've estimated the proportion of var. Rudis ancestry. And so what you'll notice when I fit these lines is that in our oldest time span from 1828 to 1920, we're seeing a very strong climb, where individuals in the west are pure var. Rudis, individuals in the east are pure var. Tuberculatus. But as we move to more recent timescales, we can see a clear shift in the distribution of ancestry. indicating that Sawyer was right. There actually has been an expansion of var rudis eastwards across the range. And this has occurred on pretty recent timescales. And instead of coloring what individual by what year they were sampled from, we can ask, how this shift might have actually led to shift in the distribution of variation across environments. So while in our herbarium samples, we actually see no difference in the distribution of ancestry between natural and agricultural habitats, when we integrate our full data set, contemporary and historical, we can see that this shift in the distribution of ancestry eastwards has also led to the preferential sorting of more var rudis ancestry into agricultural compared to natural habitats, even controlling for where they are across the landscape. So there's been this clear shift in ancestry across the range, but also across environments, right? And we can ask how that might actually relate to these fine scale signals of adaptation across the genome, right? If you remember this p-value from the CMH test, those are the regions that have high values are those involved in agricultural adaptation, right? So if var rudis ancestry is playing an important role, we might expect to see something like this, where regions of the genome that are enriched for var rudis ancestry in agricultural habitats are those that have evidence of playing a role in agricultural adaptation. So this is pretty striking to see, and I think, if Sawyer was alive, he would be pretty happy that we've shown kind of conclusive evidence that standing variation in var rudis has played a really important role in a rapid response to agricultural change. So we can not only, infer something about demographic change and demographic history from this time series data, but we can also do kind of direct observation of selective responses in allele frequency trajectories, which I think is really exciting. And a major goal, right, in population genetics is to infer kind of these parameters of selection and allele frequency change through time, but we can directly observe this. So I want to walk you through how this works. With whole genome sequencing, we can think about a single region of the genome or a single locus. And for that loci, we might have an observation of a single individual, whether or not they have the reference genotype, are heterozygous or homozygous alternate. We also know what year they were collected in. And so we can turn back to this very general population genetics model of selection, which tells us that we can estimate the frequency of some allele P at time t if we know the frequency of that allele sometime in the past, what year it was observed, and some rate change parameter S. And actually, it turns out that this model can just be rearranged to take an even more general form. That's a logistic regression model, right? So we just rearrange this, and it tells us that when we fit a logistic regression to a genotype onto collection year, that the estimated value y at x is just equal to the allele frequency trajectory, and the slope of this line is actually equal to the strength of selection. So we can apply this model to our herbarium data to infer the strength of selection and the trajectory of alleles through time. So that's what I'm going to do. First, on herbarium samples that are collected from natural habitats, right? And so if we reconstruct the trajectory of agriculturally adaptive alleles through time, you might not expect them to increase very much in natural habitats. And so here are those trajectories, just color them by how much they've increased in dark purple through time, or some of which have actually decreased in yellow. And in natural habitats, we see over the time scale of the last 150 years, they've actually increased on average by about 6%. But compared to null expectation, we actually don't see any significant evidence of allele frequency change beyond what we would expect just due to drift or random sampling. And again, not consistent with any particular evidence of selection on these alleles in natural habitats. But if instead we look at these trajectories in herbarium samples collected from agricultural habitats, we've seen actually much strong rates of evolutionary change, about 22% over the last 150 years, consistent with a rate of allele frequency change or selection of about 0.014, which is very strong over this time scale of almost a century and a half. we can also test kind of a priori hypotheses about what might be mediating these evolutionary responses to agriculture, right? And we know that, selection probably, we might expect selection not to have been constant in these environments because there's been a clear shift in how we manage weeds across this time series data. And so we can ask whether fitting multiple selection coefficients or multiple slopes to our data is actually a better explanation of the patterns we see. And so we can do that by fitting one or two multiple logistic regression lines in its kind of piecework regression framework. And we do see support for a strong shift in selection. And actually that shift falls out directly between this transition between expansion and intensification. You'll notice that this trajectory is almost completely flat in both the habitats prior to the 1950s or so, consistent with no evidence of selection on these alleles up until that time. But if instead we quantify the rate of allele frequency change and strength or rate of allele frequency change on these alleles after kind of this transition to these intensified management regimes, we're seeing an S of about 5% in agriculture, so really extreme strengths of selection over the last 70 or so years in agriculture, but also strong response to selection in nearby natural habitats. And again, I think that comes back to this idea of gene flow between agricultural and natural habitats, that as we're really applying lethal selection pressures across the landscape in agriculture, that gene flow is reintroducing these alleles into natural populations and bringing them along for the ride. So I've been kind of looking at these things on average for these alleles we've identified to play a role in agricultural adaptation all across the genome. But we can also zoom in on these alleles that we know to be causal in herbicide resistance and play a really important role in fitness. So these have been all functionally validated in Amaranthus as playing a role to resistance to acetolactase synthase inhibiting herbicides, glyphosate and PPO inhibiting herbicides. And so we can, again, do this trajectory approach. Here we might be particularly interested in the kind of confidence interval or what range of selection coefficients we might associate with each one of these resistance alleles. So we've just done a maximum likelihood parameterization of this model. And so when we look at these alleles collectively, we see even stronger selection on them than we looked at more broadly at this more broad set of agriculturally adaptive alleles. An S of about 20% over the last 70 years since herbicides were introduced. And I think what's important to keep in mind, this is averaging across a huge amount of spatial and temporal variation in herbicide use. This is also incorporating those natural populations. So this is kind of the average rate of response of Amaranthus populations to herbicides over a huge kind of spatial and temporal scale. And it's still extremely strong. For a single resistance allele, an S range is all the way up to about 34%. And so you think of, an S of 1 is lethal selective pressure. And so again, averaging across a huge amount of variation, it's pretty striking to see. And so why we're getting this, just to help develop your intuition, why we're getting these huge selection coefficients is because in contemporary timescales, there's a ton of observations of high frequency of resistance across these populations, right? So for that one with the strongest selection coefficient upper limit, that global frequency of resistance is about 35%. But in our historical samples, we're actually only observing 4 genotypes that segregate for these resistance alleles. So there must have been a huge and recent rate of change in herbicide resistance across the landscape over very contemporary timescales. So to sum up this part of the talk, we've been able to show that despite really pervasive gene flow between natural and agricultural habitats, there's still really strong evidence of rapid adaptation to agriculture. Some of that through functions in terms of growth and reproduction, especially tolerance to stress and herbicides. That demographic change, just like evolutionary rescue theory predicts, has played a really important role in the response to adaptation. And in particular, this expansion of our roots eastwards over the last 100 years has really provided the standing genetic variation on which selection in agricultural habitats has acted. and that this response has been extremely strong, especially over the last few decades, really driven by the intensification of agriculture since the 1950s. So the lab now is working to follow up on some of this work, especially in this context of temporal variation in selection. And so we've been doing some collections across the Midwest, spanning all the way up to Ottawa, down to Kansas City, where we're collecting samples throughout a growing season, before and after herbicide applications, to get at kind of the ecological timescale of response to herbicides and other selective pressures. Looking at both these resistance alleles, but the other agriculturally adaptive alleles across the genome and how quickly they're responding to different management regimes. And the hope is to follow this up year after year to get a sense of how dynamic these temporal responses really are to management strategies. Another thing we're really interested in, well, what pre-adapted Mara roots to agriculture? Like, why is this ancestry being favored? And one of the things we were thinking about is that, competition might be one thing that's been strongly selected on as these plants have invaded into agriculture, but also water availability. So these plants, in natural habitats have access to lots of water. They're growing right out of these streams, rivers, or lakes. And so what we found is actually that these agricultural populations actually tend to, on average, be more drought tolerant than natural ones, but it also correlates with longitude and ancestry. And so we're looking at what alleles underlie drought, how rapidly those respond over an extreme drought event, which we've imposed in the greenhouse, and looking at how that tempo might relate to longer term evolution to climate with our herbarium data. So kind of talked about the rate of change, but there's mechanisms like gene flow, repeated new mutation that might further facilitate the spread of resistance across the landscape. And so some of our work has been to understand those processes. And I think it's still very much related, right? We talked about evolutionary rescue model. At the beginning of the talk, I posited that new mutation might actually limit the rate at which populations can respond to environmental change. So what's the extent to which we can see that new mutation is actually limiting the spread of resistance across the landscape? Well, I was interested in testing this, and to do so, we looked at a few mutations that were very common across the landscape. So two that occur within ALS, one in amino acid 574 and one at 653. This PPO deletion at position 210. And because we have sequence data not just at that amino acid and not just at that SNP, but across the whole haplotype, we can actually use some pretty cool methods to reconstruct the evolutionary history of those genes while accounting not just for coalescent history, but recombination history. So these are some ancestral recombination graph tree sequence approaches. And so when we do that, what we can actually see is that there has been multiple mutational origins of each of these resistance alleles. So how you can tell that is, here are these phylogenetic trees of these haplotypes. The colors represent individuals or haplotypes that harbor a resistance mutation. So what we can see is that this clade of resistant haplotypes is actually more closely related to susceptible haplotypes than other resistant ones across the tree. So, right, this is the signal of multiple independent origins, and we see that for all three of these common resistance alleles. So how does that make sense? Clearly, mutation isn't limiting the rate of adaptation. Well, we can turn to kind of a very simple model in population genetics that describes the mutational input at each site every generation, right? So this is the effective mutation rate. And that's not just dependent on this parameter mu, which is the mutation rate, which we know to be very small in most species, but also the effective population size, right? Because that's the number of backgrounds those new mutations can arise on. So because I didn't want to do all the work to estimate mu, I asked, well, maybe we can try to understand this result by estimating effective population size. And so again, with genomic data, we can reconstruct that parameter, any, because we know it's inversely related to coalescent rate across those trees. And so historically, we can see effective population size has actually been very constant up until about 100 years ago. So historical NE of about 70,000. But over the last 100 years, kind of consistent with when this species became very problematic and widespread in agriculture, we see a massive population expansion. And that kind of contemporary effective population size is actually about a magnitude higher, about 80, or multiple magnitudes higher, about 80 million. So a huge number that when we plug into this equation, we actually get a theta greater than 1, right? And what that tells us is that somewhere across the landscape, a new mutation is actually arising at each site every generation. And whether or not that mutation actually is being favored in a population, is, in the context of herbicides, that selection is actually acting on it to increase its frequency, might not be the case. But it definitely, tells us that adaptation is not limited by mutational input at single sites, right? So for complex or for simple genetic architectures like single nucleotide polymorphisms, that idea might not be true. Just to complicate things a little bit more for farmers kind of looking to manage resistance evolution, I think, if it was only new mutation, we might be thinking about how we can manage these things on a very local scale. But when we map kind of these mutational origins back on to the populations and individuals where they were collected from, what we see is that a given resistance mutational origin is not only found, in one population, but, in a nearby agricultural region and even, across country borders. We can then also, on the flip side, focus in on a single population. We can see that individuals actually harbor multiple mutational origins of resistance. So it's not just, constant mutational input, but also gene flow that's really facilitating the spread of resistance. Well, that's a pretty simple architecture of resistance, right? These single nucleotide polymorphisms, but we've also done some work on complex copy number polymorphisms. And so I won't spend too much time on this, but Roundup has actually selected for increased copy number of the target gene, EPSPS. And by looking at coverage profiles around this region, we can actually get a sense of diversity or similarity in these kind of mutational origins of this amplification. So just like looking at each one of these profiles, which we've estimated for a single, each row is a different individual, you can kind of squint and see that these profiles all kind of look pretty similar. But there's a lot more heterogeneity in these profiles in Essex. And then in the Midwest, there's like clusters of individuals that look similar, but some of which look different to each other. So we can quantify this similarity parameter with the Spearman's rank correlation coefficient. And what you can see indeed that, all of these profiles in Walpole are highly correlated. In Essex, it's kind of a mishmash. They all look pretty different. And then in the Midwest, there are these populations that all seem like they might have separate origins. They're highly related within them, but very dissimilar to other profiles. So again, I think this is a case where there has been multiple mutational origins, even for a pretty complex polymorphism conferring resistance. And then there's also these populations like Essex, which seem like they're actually a collection of these different mutational origins of this complex copy number polymorphism. So in terms of kind of this monogenic trait that's really important for fitness, right, resistance to herbicides, I think it does really relate to our understanding of evolutionary rescue, where these demography, right, these massive population expansions have really facilitated rapid adaptation through providing more backgrounds on which mutations can arise. But it's not just the repeated origins, it's also gene flow that's really driving the spread of resistance across the landscape. And I think that's really a theme in the work that I've told you today. There's this clear feedback between the availability of variation that might be, spread through range expansions or population size expansion that's provided standing genetic variation on which selection in these new habitats have acted. This allows for persistence and potentially new mutations to arise, to facilitate the shift of these populations to this new fitness optimum, like intensified herbicide regimes. But now I think we're back here, right, where resistance is extremely common in these populations. We keep applying herbicides, and we're actually leading to the further spread of these resistant populations across the landscape. That's really only, leading to this feedback and innovation of the species, not just in North America, but now it's actually being done globally across the world. So with that, I want to thank you guys all for joining today. Thank all of my collaborators on this work. My lab, this is an outdated picture. Actually, I have a postdoc and a grad student now, but my lab at UChicago. And if you're interested in these types of approaches or questions, I'd be really happy to chat further with you guys today or shoot me an email.
When herbicides became increased in use,
Yeah.
there was a lot of concern about overspraying and it moving to surrounding areas.
Yeah.
So when the natural population has some increase in resistance, don't we have two possibilities? One is actually some exposure and the other is gene flow.
Yeah.
Do you distinguish?
No, we don't distinguish, right? We don't necessarily know about the history of selection in these collections that I've made. And so that's definitely something I'm looking to do now. And so some of those collections we've been that I mentioned that we've been out in the field collecting multiple times throughout the season this year, we're actually working with farmers, with extension agents and industry to get management information from those farms so we can understand the history of selective pressures. It's much harder than, though, for natural populations or populations that aren't being intentionally managed. I think some of those were pretty remote, like some of those were in state parks that, it would be pretty unlikely that I think experiencing drift from herbicides, but I can't rule it out for some of those other populations. So I think you're right, there's multiple potential sources. And I think that maybe might make some of those results even more striking. That if there is, herbicides that are even at low rates, are being, that natural populations are being exposed to, that we still see these strong signatures of adaptation is remarkable. But yeah, it would be cool to try and figure that out. Yeah.
I thought it was interesting that you mentioned like there's that one variant, the rudis one that seems to be more successful or more resistant. I'm not sure if can that hybridize with the other one but very well and
yeah, I didn't have too much time to talk about it, but they are whether or not they're subspecies or varieties, it's been debated in the literature, but there's actually continuous gene flow across the range between them. So when you look at ancestry, you can see it in this plot. Sorry, I'm just trying to find the figure. Where is it? Here. Yeah, the fact that there is continuous variation in ancestry. So you don't only have individuals that were F1 crosses, it doesn't really seem like there's any barrier to hybridization between the two subspecies. So we haven't done a lot of work looking at direction of crosses and whether we can find any evidence for incompatibilities or anything. Did I answer your question?
Yeah.
Just to follow up on.
Yeah.
Just to ask, what is it about the eastern species that wouldn't have had that genetic variation?
Yeah. What is it about the eastern one that wouldn't have the genetic variation? Some of the, there's some hypotheses about disturbance regimes, actually, and that some of these populations of rudis along the Mississippi River might have actually been pre-exposed to kind of these booms and busts of, rivers waxing and waning that actually led to selection for rapid completion of its life cycle. So this idea of what pre-adapted rudis and how is that different than what might be found in tuberculous? It's something we're looking into. One way in which we've been doing that is looking at drought. But I think, it's an open question and we're still looking to figure it out. I'm not sure. Yeah.
This is also, I think, related to the
Yeah.
question. I'm wondering if there is any like historical evidence of that variant being a pest to like maybe indigenous like agricultural practices, if there's like any historical evidence of it, and if you think that might contribute also to like the standing variation. And how like you might possibly be able to test that or know if there are samples that old or maybe if
Yeah.
there were what you think you might find.
No, that's a great idea. I think, you know, that reports are mostly of water hemp or tuberculosis being a pretty contemporary issue in agriculture. But I don't think that rules out that other, you know, that maybe before our records were very good, or that maybe that history has just been ignored, that might have been the case. Have actually been collaborating with Ben Blackman, who's using archaeological samples from some of the agricultural sediments in North America to understand the history of domestication in sunflower. I think it might be a little bit harder in water hemp because the seeds are so tiny. I don't know how much tissue it would actually have. But it's a really interesting question. Maybe at least deserves more digging in the literature. Yeah, it's a cool idea.
So it sounds like there is like an arm race going on between using herbicides versus how those populations are responding to the selection pressure. And what can we learn about in terms of agricultural application? What lessons can we draw from this kind of studies?
Yeah.
Using herbicides in agriculture.
Yeah, I mean, it's a good question. I think, you know, it's clear that we've been imposing extremely strong selection for many of these resistance alleles, right? And so if we can diversify, you know, what selective pressures these populations are experiencing, we're going to be slowing their response to selection, right? And that's not just like doing tank mixtures of herbicides, right? I think the lesson is that, you know, these populations are going to respond rapidly, no matter what way in which you try to manage them. But if you know, if oftentimes, you know, that we think, and there's lots of work that is still being done to try and think about trade-offs for resistance and costs of resistance. And if that's the case, then diversifying how we're managing these weeds should mean that it's slowing the response to any one selective pressure. I think there's also to be said something about the scale at which management needs to be happening, right? Which I alluded to a little bit. Like if it's only, you know, new mutations that are driving the spread of resistance, that means it's really the local kind of on-farm practices that are, you know, facilitating whether those mutations are selected for and spread. But when there's migration, kind of spreading these resistance alleles across the landscape from your neighbor or from the next county over, we maybe need to be thinking about more kind of legislation or kind of county level practices that are thinking about like hygienic, you know, sharing of machinery. I mean, we can't really stop, you know, ducks from eating these plants and, you know, pooping on your fields, but there has to be, you know, some, I think, multi-scale level approaches in which we're thinking about how to prevent the spread of resistance. Yeah.
So given these high population sizes
Yeah.
and the strong selection, what do we know about reversion back to susceptibility? Has anyone done sort of experimental work in that?
I mean, I think the hope is that there's these costs of resistance alleles, and if you stop applying herbicides, then you're going to lose those resistance alleles, right? And some work, like having kind of nearby refuges to keep introducing these susceptible alleles, can work to prevent this slow spread of resistance. But I think what we're seeing for many herbicide resistance alleles is that it doesn't seem like there are consistent costs, right? Selection in natural habitats where herbicides probably aren't being applied, isn't such that these alleles are immediately lost in those populations. And so I think that tells us that, I think it tells us that the reversion to susceptibility might not necessarily be the case. I mean, we don't see a lot of standing variation for resistance, which kind of goes against that idea. But I think whether or not there are strong costs to resistance alleles is still kind of an open question, and there definitely is more work to be done on it.
curious about the amaranthus introductions, let's say in Europe.
Yeah,
And here you will have a much narrower gene pool that's being
Mhm.
transferred.
yeah.
Can you correlate what's happening there with what's happening here?
Yeah, that would be a really nice future direction, and especially where, there's actually variation too in management strategies. Like in Germany, they're not applying herbicides. And so like how parallel are this response and adaptation to agriculture in different parts of the world? And how has introduction history and these bottlenecks actually limited, maybe the input of new mutation or, compressed the standing variation in which these populations can adapt to agriculture. So yeah, it's, I think this is all happening in real time. Like this invasion to Italy and Spain is just in the last couple of years. And so it's definitely something, I was talking with Ramon Leon, he's working on some of this right now, even in Amaranthus. And so it's some stuff that people are working on and be really cool to follow up on. Good idea. Yeah, one in the back.
So could you say something about how the different mechanisms of action of herbicides may impact selection of seed?
Yeah, I kind of totally glossed over the physiological kind of mode of action. Let's see, maybe I'll just go back to this slide. So there's these different classes of herbicides, and some of these different classes even so some of which target, genes that are involved in protein production, right? So if you stop a plant from being able to produce a certain protein, it'll die. Other ones are targeting photosynthesis or auxin signaling, right? So there's all these different ways in which you can screw with the plant's growth and reproduction, right? And so these are what, folks have figured out and developed new herbicides based on. I don't know how much I can speak to how those different classes impact or facilitate adaptation other than, very broadly, I think that, glyphosate or Roundup herbicides, a lot of you guys have heard of with Roundup Ready crop regimes, they hypothesized that resistance would not evolve because there was a bunch of protein modeling where they looked at every amino acid residue and thought, well, if you had a non-synonymous substitution there, it would be too costly and that mutation would not persist to allow for resistance, like that plant would die anyway. But what they didn't expect, was that copy number amplification might arise, right? And what they didn't also maybe think about was that mutations all across the genome, each of which have small effect, might contribute to resistance. So some of the work we're doing is trying to understand what the how much what the exact word I'm looking for, but what genetic architecture might underlie some of these different responses to herbicides, and maybe some are constrained to these particular SNPs, but maybe for some other ones, there's many more ways in which these plants can evolve to herbicide use. So this is some of the work we're doing right now. I don't think we actually have a super strong idea about, what's limiting the rate of adaptation to those different classes of herbicides, at least from this population genetic perspective.
Do you have historical data of what type of herbicide was applied at each one of your regimens?
Yeah, it's a great thing, a great idea, and it's like you always go back and think about how you could have done things better on your paper. But I had this figure where we kind of estimated a single selection coefficient for all of these, we did it collectively for all herbicides, but also each for independent ones. But we did so since kind of this intensification period. But really, these herbicides were actually introduced in sequence, right? There was a single herbicide that was rolled out in 1956, and then other ones which were introduced in 1970, and then others in 2000. And so we could look at that. And the USDA has also been tracking herbicide use at the state and county level over the last 30 years. So some of our ongoing work right now is to incorporate that spatial and temporal heterogeneity in herbicide use to understand how that actually relates to allele frequency change through time. Yeah.
So I have a question in the chat.
Yeah.
From David Andau, who
Okay.
is the department head in applied ecology. And he says, very interesting work. One, there are many species of weeds in these agricultural systems, such as butylon, chenopodium, and other species of amaranthus. What distinguishes rudis from these others?
I guess in terms of ecological strategy, I think one reason it's so problematic is that a single individual can produce millions of tiny seeds. Like I think the upper estimate's 5 million. And that those seeds can remain dormant in the seed bank for like 15 years. So it's a huge problem, you know, in that populations now have cross resistance to every chemical mode of herbicide. So if maybe in part because of these, you know, dioecious outcrossing mating systems, these large population sizes. But now that we've kind of maybe outgrown some of the typical herbicide management approaches, that, you know, farmers are having to get in the field and do hand weeding. And if they don't, this is going to be a persistent problem year after year after year because of that seed bank. So I think that's a big part of why it's so hard to control in these settings.
On that note, you briefly mentioned the sex determination.
Yeah.
Have you been able to look at if those kind of subdioecious individuals are increasing or decreasing? Is there selection to maintain dioecity so that they can keep evolving resistance better?
No. I haven't looked at it. Yeah, it's kind of a very recent result that I took a while to wrap my head around. But yeah, these subdioecious individuals, like we don't even know the frequency at which they occur in the wild. I mean, from genetic data, we can see like it's like a frequency of about 5%, but I've never seen them when I've grown them. And so I'm like, you know, we're going back, we're growing plants in the wild, we're taking a very careful eye to these things. But I think it would be really cool to learn more about how that's interacting with kind of adaptation in the system.
Okay, so we have a little discussion.
Okay.
About reversal. right?
Okay.
And if you have ideas and stuff, Ramon Leon says, we have never seen herbicide resistance reversing susceptibility. We now have multiple resistance with plants exhibiting resistance to 5, 7 mechanisms of action. So, I mean, you might want to elaborate on what you think.
Yeah. I mean, I guess there's multiple ways in which reversal can happen. You know, one is which like a second mutation or like occurring, which I think is probably unlikely. Although what we're seeing here is that maybe that's not impossible because there seems to be so many individuals across the landscape, and that, you know, one mutation might arise somewhere. But like Ramon said, yeah, like once a resistance allele fixes, I mean, the thing is that most of the populations that I've looked at, I've never seen a population that had fixed resistance. And I think part of that is migration, right? So I guess it's like unclear what reversion actually refers to. Is it that there's your hope, you know, is it driven by costs of these resistance alleles? Or is it the kind of reintroduction of susceptible alleles into the population through migration? So I think those are kind of different mechanisms that might reintroduce susceptible alleles.
And if you're increasing copy number, the probability of having reversal mutations on all of them is probably 0, right?
Yeah, yeah.
Any other questions?
There's one more friend.
Well, pardon me, but this is a silly comment. When I look at a confidence interval of -infinity to +infinity.
Oh, yeah.
It occurs to me that that's 100% confidence interval.
Yeah, those ones we like have no observation. There's such low frequencies of resistance in the contemporary and none in things. So we can't really say anything about selection coefficient. Nice point.
All right, I guess with that comment, we are, thank you.